You're now at the lab with Cold Spring Harbor Laboratory. My name is Mark Prasad, and this week at the lab, Outmuscling cancer. Diseases aren't cured overnight. It takes years, often decades, for biomedical breakthroughs to materialize. But every now and again, there's a turning point, a moment when years of hard work starts to pay off. CSHL professor Christopher Vakich may be rounding such a corner.
We can't see the future, but every successful drug has its origin story, and studies like this are the soil out of which new drugs are born. So it's a fundamental discovery. It's learning one of the most important molecules in a disease that kids are dying of.
The disease he's talking about is rhabdomyosarcoma, or RMS, a form of cancer that mostly occurs in children and teenagers. The molecule is a protein called NFY. We'll return to that shortly. But first, let's talk about RMS and how the Vakich lab came to study it.
The name rhabdomyosarcoma refers to the similarity of the tumor to muscle stem cells. There is not a lot of research dollars directed at this cancer, and that's a big part of how we got into this. There are families that have lost children or spouses to this disease on Long Island, and they all came together and funded us to try to find a new therapeutic strategy for this disease.
About 10 years ago, the Vakich lab had a radical idea. If RMS cells are essentially muscle stem cells that never transformed into muscles, maybe they could stop the cancer's growth by completing the cell's transformation. It took years to develop the genetic screening test needed to pinpoint the molecular machinery that would cause the RMS cells to change. But finally, NFY emerged. Vakich's team used CRISPR to target NFY inside the RMS cells, and that's when it happened.
These tumor cells, which can't contract, now become a muscle cell that has the whole contraction machine. It's like a fork in the road, and the cell has gone down one path, but now it can't go back to this multiplying state because all of its energy and resources are devoted to contraction.
A fork in the road for RMS cells, and just maybe a turning point for RMS research. And it all happened here at the lab. Thanks for listening. To hear more fascinating science stories like this one, please subscribe wherever you get your podcasts. And visit us online at cshl.edu. For Cold Spring Harbor Laboratory, I'm Mark Prasad, and I'll see you next time at the lab.